Welcome to Class 9 Biology, chapter The Fundamental Unit of Life. So far you have studied about different animals and plants in your biology lessons. We studied the characteristics of plants and animals. We studied the grouping of plants and animals based upon their characters. Now, we are learning something which is deeper. That is the construction of plants and animals, with what they are made. So the topic was introduced in your 8th class, the living things with what they are made up of. So here in this lesson, the fundamental unit of life, we are going to study what is the fundamental unit of life. What is its construction? What are the various structures that make up the living things? How they are? What are the functions? And what are their characteristics? All these things we are going to study. See, this is a wall. It is a single unit, single entity. Yes, we see the wall. If the wall is plastered, we do not know with what this wall is made. The same wall without any plastering. So some part of the wall, it lost its plastering. From there, you can see that the inner things of the wall. These are the bricks. By this you can identify the wall is made up of bricks. So a wall is made up of some bricks. 100 bricks, 200 bricks, 500 bricks. So brick is a unit which makes up the wall. In the similar fashion, the living things are also made up of some basic units, fundamental units. So this thing was observed by a person called as Robert Hooke. In the year 1665, Robert Hooke observed the section of dead cork cells. Cork cells. So cork is nothing but wood, the dead cells, dried up wood. He has taken a very thin section of the wood, and that section he observed under a microscope. There he found small, little rooms-like structures under the microscope. He found little room-like structures, small rooms, little room, little rooms. These little rooms in Latin, they are called as cells. So he called these small rooms as cells. So from there the word cells has come into the biology. Thereafter, people started studying cells. What is the importance, need or necessity of studying a cell? Because instead of studying the various characteristics of an organism, if you study one cell of that organism, you can understand many things. So there, the cell biology gained the importance. Cell biology. So the construction of a cell, the composition of a cell, various kinds of physical, chemical, biological, biochemical, biophysical reactions takes place at the cellular level, the movements of molecules at the cellular level. If we study all this, then they will be very helpful in various fields of application. The study of a cell helps the scientists to design good medicines. Here they cannot study the complete organism. It may or may not be useful. They study the particular cells, the behavior of these cells under certain circumstances. So in this way we can design the medicines. We can find the preventive measures for various diseases. And we will come to know so many things and how to control or prevent diseases like even cancer, how it is happening, the reasons behind a change or a disease or a disorder or a metabolic disorder. All these things can be understood by studying the cells. So this is the importance of the studying the cell. So cell is the fundamental unit of life. In the first part, in the first session of the lesson, we explored the fact the cell is the fundamental unit of life. Cell is the fundamental unit. Cells were first explored by Robert Hooke, that is from a dead cork cell, that is from that wood which belongs to plants. So later the ideas developed that both plants and animals, they were made up of these cells, the fundamental units. So an organism is composed of so many tiny units called as cells. And how are these cells? Say for example, in this wall, there are so many bricks. The bricks, they made up the wall. But are the bricks functional? No, they are not functional. Just they are physically a part of the wall. They are not functional. But whereas if you see a living organism, either a plant or an animal, they are made up of cells, the fundamental units, and these units are functional. Means there are so many activities, metabolic reactions takes place in each and every cell. Each and every cell, it does lot of things, prepares new materials, gets energy from the materials, nutrients, utilizes the energy to produce new materials, removes the waste materials, removes the waste gases. In this way, so many activities or processes going on each and every cell. So if each and every cell carries out this, and if each and every cell is healthy in our body, then we are healthy. If the functioning of a cell is disturbed, then the functioning of the body is also disturbed. So cell is the functional unit. So we can understand that the functional unit is the fundamental unit. It is the fundamental unit. At the same time, it is a functional unit as well as structural unit. So a cell is a structural and functional unit of living organisms. So that's what we here learned, that a cell is a structural, means it helps in giving a structure, as well as structural and functional unit of living organisms. So Robert Hooke did that. He identified with an experiment in the year 1665, long ago. But now you got a doubt, you doubted. Is it true? Or the living organisms and their parts are made up of cells? I wanted to confirm. Yes, you can confirm it. You yourself can carry out an experiment to confirm. You yourself can witness the living things, living organisms are made up of cells. How could you do that? You can do that by very simple experiment. You need any plant part, a vegetable kind of thing, to make a thin section. For a better and easy activity, you can choose onion, and you can take the peel of onion and you can prepare a temporary slide. By viewing this slide under a microscope, you yourself can witness the living things are made of cells. Let us see that activity. Now let us look at the activity, how we can prepare a temporary slide of onion cells, that is to personally witness the living things are made up of cells. So what do we need to do this activity? We need an onion bulb, because as I told you, easily we can prepare a temporary amount of the plant cells. We can easily see that. So the onion bulb is fleshy, it contains water. That means the leaves of onion bulb, they contain water. So by that they will be having good visibility, and even the size of the cells are also bigger to see. So here we need a forceps. With the help of this forceps, you can take a very thin peel of the onion. So immediately you take the peel, it gets dried. There is a chance of getting dried, or it may be folded. If it is folded or dried, you cannot make a good slide. So what to be done? Soon after you take the thin peel, immediately the peel should be placed in a watch glass. So what does this watch glass consist of? Water. Take some water into this, and in the water you place this small peel which you have collected. It will prevent the folding or drying of the sample you have collected. So here the samples are there. Now how to take that sample to this slide? So there you need a slide, a glass slide, which is used to view tiny things under a microscope. So this is the glass slide, and from here you have to transfer this sample very carefully with the help of a paint brush. You need a paint brush, camel hair paint brush, so which you use for drawing, painting. That paint brush you can take it, and with the help of paint brush you have to transfer the content here. So you have to transfer the sample here. Now the plant cells, they have very pale color. Because of lighting in the microscope, you may not be able to see the cells properly, because the cells are in very pale color. Even the cell walls are in very pale color. You may not be able to distinguish the cell walls and the cells. So what we are doing, we are making the color of the sample a little brighter for a better vision. How can we do that? We can add a drop of saffronin color. If you don't have saffronin, you can take some red Indian ink color, red Indian ink, Camlin red ink you can take, and do not put the concentrated ink here. It will become too red, so by that you cannot see anything. Dilute it with water and put a drop of ink onto this sample, or put a drop of this saffronin solution onto this sample. Now carefully cover it with a cover glass. Cover glass is another glass slide which is very thin and small. This is the glass slab which is bigger and thick. Thin flake like glass slide will be there, that is the cover slip, and with the help of a mounting needle, carefully place the cover slip over the sample. So your temporary mount of onion peel is prepared. Now the next thing is to observe this under the microscope. So you need a microscope. Which microscope we need? We need a compound microscope. Compound microscope. So this is a compound microscope. So this compound microscope let you observe the tiny things like cells. If you place this slide made with onion peel at this position, if you adjust the light by adjusting the mirror, if you focus the light to this hole, and if you adjust this objective and lens, and here there are knobs for fine tuning, coarse adjustment and fine adjustment. If you do the adjustments, then you will be able to see a clear picture of cells exactly like this. You find the cells are rectangular, somewhat rectangular like this, elongated cells. So why I have drawn a circle? Because that is the view. Here there will be a circular lens. So this is the vision you get. And you will be able to see dark bodies at the center of these cells. Those are nothing but the nucleus. If your microscope is of high power, then you will be able to see the nucleus. If it is of low power, you may not be able to see the nucleus, but you will be able to see the clear distinction between cell to cell, that is the cell wall. Because you have added some color. Because of the color, you can see the cell walls are red, as we have added saffronin. And we can see the brick-like arrangement in the onion peel. You can do this experiment with the help of a leaf peel, betel leaf, any other leaf, any fleshy leaf like aloe vera you can take. So you can cut the part of a plant The leaf part you can observe, even you can observe stomata. So this is the way how we prepare a temporary mount. So why we are doing this activity? In the beginning I told Robert Hooke observed the cork cells and he found some room-like structures. So you're also doing the same thing. That is for your personal experience. And you yourself are experiencing, visualizing, seeing the cells by doing this activity. So that is with the discovery or usage or invention of a microscope. So this was the great invention or discovery in biology, in the field of biology, because this microscope helped the scientist, it helped the mankind, or it revealed two important facts to the mankind. What are they? The first thing is living things are made of cells. So this is the first thing which we discussed so far. So what is the second thing? Certain living things have only one cell throughout their life. So the first thing is living things are made up of cells. Every living thing is made up of cells. Certain living things may be made up of thousands and lakhs of cells, certain millions of cells, certain trillion of cells. But here, certain living things, they have only one cell. The organism itself, one cell. Example. So by the invention of this microscope, scientists observed different kind of things under the microscope and they found that certain organisms like amoeba, paramecium, chlamydomonas. So all these are unicellular organisms. All these are organisms having one cell throughout their life, and they categorized all these organisms into unicellular. They called them as unicellular. Uni means one. Then what about the other organisms, other plants, fungi, animals? These are called multicellular. Multicellular. Multi means many. Cellular means cells. So unicellular, multicellular. There is a classification. Here we have to notice one more important thing. So first thing is living things are made up of cells. Second thing is certain living things, they have only one cell throughout their life. And other organisms, they have number of cells, so many cells. Here what is the third thing we have to notice, that multicellular organisms do not have same kind of cells. Multicellular organisms are made up of many cells, but all the cells are not alike, not similar. Multicellular organisms, what do you call it as? Differentiation of cells is found in multicellular organisms. What is this differentiation? We are made up of cells. I am made up of cells. The cells that are making up my tongue, the cells that are making up my skin, the cells that are making up my bone are not alike, not similar. Even though a living organism is made up of number of cells, all the cells are not alike. The cells that make up a living organism are differentiated. That means they have different shapes as well as different functions. You call it as differentiation. Let us see. So let us talk about the cell organelles. The first and the most important cell organelle is the plasma membrane, and this is also called as cell membrane. So plasma membrane or the cell membrane is the outermost covering of a cell. It is the outermost covering which separates the cell and its constituents from the external environment. So you are able to tell or say this is a cell. How can you tell? By identifying its border. This is the outermost covering. By identifying its border. What is this border? This is the plasma membrane. So all the constituents, the jelly-like cytoplasm, nucleus, other cell organelles are enclosed by this cover, plasma membrane. So what are its functions? Plasma membrane, it has two major functions. Protection, it protects. And the most important function of the plasma membrane is permeability. Even though here the protection also done by the permeability. What is this permeability? Means allowing substances to get in and get out. You see any office, any corporate office, any government office, you will find some security outside the office. So you see that even houses, apartments also have some watchman or security. So if somebody wanted to get into the office or into the apartment, the security people will stop. Who are you? Where are you coming from? So they find out whether the person is the right person to send inside, or he may be a dangerous person not to send him inside. So the security, they will check. If everything is fine, they will allow. In the same way, materials cannot enter into the cell directly. If it happens so, the cell gets damaged. So the cell is protected by the selective permeability nature of the plasma membrane. So what is this permeability? Selective permeability. And what it is allowing? Actually, what are the materials that travel inside and outside the cell? What materials will go inside the cell and what materials will come outside the cell? So what are the materials? Basically, three kinds of materials travel across the cell membrane or plasma membrane. One is gases. The second is liquids like water. And the third is minerals and other nutrients. Minerals and other substances. So we write other. So first, gases. What kind of gases? They travel across the plasma membrane into the cell and outside the cell. Two main gases: oxygen, carbon dioxide. So cell, it carries out various activities. So for the activities, it needs energy. The energy is supplied to the cell by the process of cellular respiration. Means respiration takes place inside the cell. So for the respiration, what gas is required? The oxygen is required inside this cell. And during the respiration, what gas is produced? Carbon dioxide gas is produced. So the excess of carbon dioxide inside the cell is dangerous to the internal environment. So the excess carbon dioxide has to be expelled out, sent out of the cell. So the CO2 must go out and oxygen must enter the cell continuously. It's a continuous process where a cell takes oxygen, leaves the carbon dioxide. But how it happens? How the transfer of gases takes place? So the transfer of gases takes place between the external environment and the cell. It is done by a process called as diffusion. Diffusion of gases. So how this diffusion works? On basing what principle? The diffusion, it works on concentration. Already you studied about the diffusion process, even in physics, even in your chemistry part also. The best example you studied. See, for example, if you light a incense stick, agarbatti, and if you place it in a corner, slowly you get the smell throughout the room uniformly spread. Means the smell molecules, they spread uniformly throughout the medium. Uniformly means from a place of higher concentration to place of lower concentration. If you take a beaker of water and put a drop of potassium, if you put a drop of ink or a few crystals of potassium permanganate, slowly the color spreads throughout the beaker uniformly. means the color molecules spread uniformly, that is from a point of higher concentration to point of lower concentration to make an equilibrium. So in the same way, inside the cell, whatever the oxygen is there, this oxygen is being consumed by the cell for various activities. So here is also oxygen, here is also oxygen. Inside the cell, the oxygen is being used up. So the concentration of the oxygen falls down inside the cell. Oxygen, it diffuses from higher concentration to lower concentration. Because outside the cell, the concentration of oxygen is high. Inside the cell, the oxygen concentration is less. Why it is less? Because it is being used up in the process of respiration. In the same process of respiration, CO2 is being produced. As the oxygen is used up, CO2 is produced inside the cell. If you see the concentration of CO2 inside and outside, the outside environment is having less carbon dioxide compared to the internal environment of the cell. So the carbon dioxide, it moves from cell to the outside of the cell. In this way, the exchange of gases takes place, which is based on the process called as diffusion. So what does this diffusion helps? How the diffusion helps? It helps in the exchange of gases. And how it works? It works on the principle of concentration. Inside the cell, as the energy is required, oxygen is used up continuously to produce energy. As the oxygen is being used up, the concentration of oxygen is less in the cell compared to the outside environment, so the oxygen is diffused into the cells. As the cells are using up oxygen to produce energy, in the same process carbon dioxide is produced. So the percentage of carbon dioxide is high inside the cell compared to the external environment. So this carbon dioxide diffuses outside the cell. The same happens. If you take amoeba, is a single-celled organism. It is having only one cell. Then how does this amoeba respire, get the energy? Amoeba takes the oxygen directly into the body, releases the carbon dioxide outside the body. How it happens? That is by the process of diffusion. The same, if it is amoeba or if it is our cell, whatever it may be, the process is same. The exchange of gases into the cell and outside the cell takes place by the simple process called as diffusion. So diffusion of oxygen and carbon dioxide. So now let us see, the same diffusion is also applied to water also. Water also follows the same diffusion. Water also should enter and leave the cell. Cell carries out so many activities. Cell not only require oxygen, cell also requires water. See, when you have traveled in sun, hot sun, you will sweat, means you lose water from your bodies. From where does the water comes from? That is from your body, from your cells, the water comes out. Because of the heat, to keep your body cool, the water comes out of the cells. Now your cells are dehydrated. Then it triggers one urge in your mind, thirsty. So you feel thirsty, means you wanted to drink some water. Why? Because the water is needed for the cells to get rehydrated. If the cell is dehydrated, cannot function properly. Right? So that is the reason you need to drink water. So cells, they lose water, they gain water. But how the water travels through this plasma membrane? So water also, the same principle based on the concentration. Inside the cell, here is the water. Outside the cell, also there is water. But how the movement of water molecules takes place inside the cell? Inside the cell, if the concentration of minerals, whatever the cytoplasm is there, that cytoplasm concentration is more, then the water molecules move inside the cell. When there is more amount of water here compared to the outside environment, the concentration of the external environment is higher than the cell, then the water moves out. It all depends upon the concentration. So what is this concentration? Let us understand this with the help of an example. The next cell organelle which we are going to discuss is a nucleus. Nucleus is the center of the cell. It is the controller of the cell. We have seen how to prepare a temporary mount of onion peel. There we discussed about staining the cells, that is for a better vision, for a clarity. If you use a high power microscope, objective, that is compound microscope, then you will be able to see the onion cells, that is you can see the cell membrane at least, and at the same time you can find something else inside the cell, that is the nucleus. So if you use certain kind of staining agents like iodine, or you can use safranin or methylene blue, because certain colors, they colored certain parts of this cell, especially the nucleus. You can find some dark bodies inside the cells. Of course, you may not be able to see what is there inside this dark body clearly. You will find these dark bodies as dots, that's all, by using an objective compound microscope. If you use an electron microscope, you will be able to see what is there inside the nucleus even. But till the level of compound microscope, you are able to find out or locate the nucleus. Now let us see the structural features as well as the functional features of nucleus. First, let us talk about its structure. Nucleus is a round dark body which is having two membranes, outer membrane as well as inner membrane. So two membranes are covering the nucleus. The membrane is called as nuclear membrane, inner nuclear membrane, outer nuclear membrane. And what does this nucleus consists of? Nucleus consists of a thread-like entangled mass of material called as chromatin. Nuclear material, chromatin. So what is this chromatin? Is it a ball of thread? What is this chromatin? This chromatin is made up of two important materials called as DNA and protein. So what is this DNA? Deoxyribonucleic acid. This is our genetic material which will decide the characteristic features of an organism, which is very, very important in determining the features of an organism. That is the DNA. Its full form is deoxyribonucleic acid, and protein. These two together they form the chromatin. Chromatin is the entangled thread-like mass which is found inside the nucleus. And the nucleus is bound by two membranes, the inner membrane and the outer membrane, together called as nuclear membrane. And of course, the nucleus contains one more dark body called as a nucleolus. Now, what is this chromatin? We studied in the lesson of cell division, mitosis and meiosis, we studied about chromatids. Then, what is the relation between this chromatin and chromatids? When a cell is not dividing, it is not in the stage of division. In such cases, you find the genetic material as chromatin, the mass of entangled thread-like material, chromatin. But when the cell is about to divide, when the cell is ready to cell division, or when a cell is in the phase of cell division, you find the chromatin in the form of chromosomes. Rod-like structures, chromosomes. The chromatin, it becomes two chromosomes. That is during the process of cell division. So whether it is a chromosome or a chromatin, it consists of these things like DNA and protein. DNA is the genetic material. It will consist of all the information about various features of your body. So what is this DNA is composed of? What is the fundamental unit of DNA? Genes. Genes are the fundamental units of DNA. So genes, the composition of these genes, they make the DNA. Just imagine the DNA is a ladder. So all these parts of the ladder are genes. Our genes, they pile up, attach it to one another, they make a long helical thread-like structure called as DNA. So this is the structural part of the nucleus. Then what is the function of the nucleus? What is the function? Functions: control. Second thing, coordinate. The cell consists of various organelles. It consists of mitochondria, it consists of lysosomes, Golgi bodies, and different kind of things. So these things, they have to do different tasks, but they don't do the tasks aimless. That means unnecessarily the cell organelles do not prepare anything. If a cell requires something, if your body requires something, the cell will prepare the material. But who will give the instruction to the cell organelle to prepare that material? How much material has to be prepared, produced? So that is decided by the nucleus. So some oil has to be produced on your skin, the skin cells. The oil is produced by the oil glands. The oil gland consists of cell. The cell consists of cell organelle which will prepare the oil. But the cell organelle, how it will know when to produce the oil, how much oil has to be produced? It is based on the instruction given by the nucleus. Of course, sometimes the nucleus, it gets the information from other cells. So the nuclear membrane will be having some pores for the exchange of information from the nucleus to the cell organelles. So the information is carried from nucleus to the cell organelles, parts of this cell. There is some communication. So this way, the nucleus, it will control the other organelles. It will coordinate. For example, if the material required is produced in excess quantity, it will initiate a feedback mechanism. Convert the excess material to some other material, store it somewhere. In this way, the coordination and control of the cell is achieved by the nucleus. And moreover, nucleus plays a very important role in cell division, mitosis, meiosis. When the cell division takes place, the chromosomes, they are divided in a specific manner in case of a mitosis, in case of a meiosis. So by that, the genetic material is transferred to the newly formed cells in a proportionate way, proper way, not missing the genes. So by that, the abnormalities may arise. So that is not happened, which is achieved by the nucleus. So it plays a very important role in cell division. And at the same time, it controls the activities or functions within the cell. It controls all the other cell organelles and other cell activities. So what we are discussing about, all about the multicellular organisms. Multicellular organisms, they have a well-defined nucleus in their cells. In certain unicellular organisms, in not all unicellular organisms, I mean in certain unicellular organisms like bacteria, they do not have a specific nucleus. Unicellular organism like amoeba, it has got a specific nucleus. Its nucleus is clear, distinct. The nucleus is covered by nuclear membrane and it is having the similar nucleus as what we have. But certain unicellular organisms like bacteria, they do not have a specific nucleus. Then how do you achieve their cellular functions carried out? If this is the bacteria, then how the control of the other organelles takes place and how the division of the cell takes place. So there they find the nuclear material is grouped at some lump, some mass, without having a proper nuclear membrane. So without having a proper nuclear membrane, the nuclear material is gathered with some proteins. You call it as nucleoid, not nucleus. You cannot call it as a nucleus, but you call it as nucleoid, which is found in primitive organisms. Certain organisms which do not have a nucleus are called as prokaryotes. Prokaryotes. Bacteria. The organisms that do not have a specific or a distinct nucleus, but they have nuclear material directly in the cytoplasm, you call them as prokaryotes. The organisms that have a distinct nucleus with a double layered nuclear membrane, you call them as eukaryotes. Eukaryotes. So prokaryotes and eukaryotes is the division of organisms based upon the presence of distinct nucleus in their cells. So let us talk about the endoplasmic reticulum. In simple terms, it is called as ER. If you see an eukaryotic cell, you will find the endoplasmic reticulum in the form of elongated, oval or oblong bags or vesicles. It is in the form of bags or vesicles. See here that they have some channels. They look like a network of channels that they are extending from the nucleus towards the plasma membrane. So they are forming some kind of channel. They are forming some kind of way. Now let us see, just we have seen the structure. Even though they varied in their shape, size and their orientation in different cells, but they form a network which is called as endoplasmic reticulum. Basically, we find two different types of endoplasmic reticulum inside a cell. One is SER, the other one is RER. What is this? SER stands for smooth endoplasmic reticulum, and RER stands for rough endoplasmic reticulum. So here it tells the nature of it, smooth and rough. What is smooth? What is rough? The endoplasmic reticulum, certain endoplasmic reticulum, it consists of small granule-like particles called as ribosomes. Ribosomes. If the endoplasmic reticulum consists of these ribosomes, if it consists of these ribosomes, it is called as RER, rough endoplasmic reticulum. If the endoplasmic reticulum does not contain any ribosomes, it is called as smooth endoplasmic reticulum. Means the endoplasmic reticulum with ribosomes, rough endoplasmic reticulum, without ribosomes, smooth endoplasmic reticulum. So structurally they are varied. Of course, everything is called as endoplasmic reticulum. But there exists only the structural difference or any functional difference between the smooth endoplasmic reticulum and rough endoplasmic reticulum. Yes, there is a functional difference too. The rough endoplasmic reticulum, it involves in the function of protein synthesis. Protein synthesis. Smooth endoplasmic reticulum, it involves in a function of lipid synthesis. They manufacture lipids. Smooth endoplasmic reticulum, it manufactures the lipids. Rough endoplasmic reticulum, it manufactures the proteins. The manufacturing, the preparation is termed as synthesis. So this is achieved by this SER and RER, what they are making, lipids and proteins. Why they are needed? In the previous session we studied, when we were studying the plasma membrane, we learned that plasma membrane is made up of lipids and proteins. So for the construction of the plasma membrane, lipids and proteins are required, which are prepared by endoplasmic reticulum, which is termed as biogenesis, preparation of synthesis of lipids and proteins for the formation of cell membrane is termed as biogenesis, takes place in the endoplasmic reticulum of the cell. It is a major function achieved by the endoplasmic reticulum. Of course, various proteins that are synthesized in the rough endoplasmic reticulum, these proteins are converted to very important biochemical molecules like enzymes and hormones, which play a very active role in the control, coordination, development and metabolism of an organism, enzymes and hormones. So the enzymes and hormones are even chemically they are proteins which are synthesized here. So that is the importance of this endoplasmic reticulum. And what is the other function apart from the synthesis of materials like lipids and proteins? What else it does? So it acts as a channel for communication. So what has to be communicated? From where to where it has to be communicated? The communication must reach from the nucleus to some other cell organelle, a mitochondria. So the information should pass through some material, some agent materials to reach this place, and this endoplasmic reticulum, it acts like a network channel for communication, for passage of materials from one place of cytoplasm to the other place of cytoplasm. So that is the other important function carried out by this endoplasmic reticulum. Here we find some other important function apart from the two functions which we discussed in certain kind of vertebrates. Like in humans, the liver cells, they contain the endoplasmic reticulum, which will detoxify the poisons and harmful drugs. So that takes place in the endoplasmic reticulum. So the endoplasmic reticulum, it provides a surface for many biochemical reactions, very important biochemical reactions. They takes place on the surface of this endoplasmic reticulum. So in this way, the endoplasmic reticulum, it helps a lot in the biochemical activity of this cell. That is, in the production of useful materials, say whether proteins in the form of enzymes or hormones or other metabolites, which are very essential for carrying out some very important reactions inside the cell. So that is the prominence or importance of the endoplasmic reticulum. So next we are going to talk about another cell organelle, that is Golgi complex. Sometimes it is also called as Golgi apparatus. Why are we using this apparatus and the terms complex for this cell organelle? So, and how it got the name Golgi? What does it mean? So these organelles were first described by a scientist called as Camillo Golgi. So they're called by the name Golgi in honor of him, and they are called as complex or apparatus because of their structure and function. If you see them, they are like vesicles arranged parallel to one another and having some connection. This vesicle is connected to this. These are called as cisterns. So this is at some point connected to endoplasmic reticulum, Even in its function, it is having a connectivity to the endoplasmic reticulum. What is the connection? What is the function of endoplasmic reticulum? Endoplasmic reticulum function is production of proteins and fats, or production of biochemicals, enzymes, hormones. These kind of things are produced here. See, if you see a factory, the goods are produced. So after production, the goods are not taken to the customers directly as it is, as they are produced. They are to be packed properly. Otherwise, it gets spoiled. It cannot be transported properly if there is no proper package. Right. So, in the similar fashion, how the materials produced in a factory are properly packed, sealed, and properly dispatched, in the same way, the dispatching of the materials which are produced in the endoplasmic reticulum is done at this Golgi apparatus. So the main function of this Golgi apparatus is to pack the materials that are prepared by endoplasmic reticulum and dispatching these materials. Sometimes the Golgi apparatus converts the materials which are formed in the endoplasmic reticulum. That means certain complex sugars it will convert to simple sugars, and these materials will be properly packed. This Golgi apparatus, it forms the vacuoles which are packed with materials, and these vacuoles, they travel to some other place where they are required, and they are responsible for formation of lysosomes also. If some other materials or waste materials or harmful materials are converted to some other form, they are to be dispatched or disposed out of the cell. So they are packed in the form of a lysosome. In this way, the Golgi apparatus, it takes the function of dispatching the materials which are synthesized by the endoplasmic reticulum. So that is the reason it is connected even to this endoplasmic reticulum. So as it is having connection with these endoplasmic reticulum itself, and it is consisting of so many things together, you call it as a complex, or otherwise you call it as an apparatus. So that is the main function of the Golgi apparatus. Lysosomes are the small, round, vacuole kind of things present inside the cells. So what is this a lysosome consist of, and what is the function of it? Structurally, the lysosome, it consists of certain digestive enzymes which were made by the rough endoplasmic reticulum. So rough endoplasmic reticulum makes certain digestive enzymes which are passed to the Golgi apparatus, and the Golgi apparatus, it packs these digestive enzymes into a sac called as lysosome. So why the Golgi apparatus is making these lysosomes with digestive enzymes? What is their function? The main function of these lysosomes is to digest the unwanted materials inside the cell. What are these unwanted materials? Unwanted. What does this term refers to unwanted in this situation? In this circumstance, they are worn or damaged cell organelles, 2. Any viruses or foreign particles, bacteria, anything that enters the cell. So these kind of things, if they enter into the cell, they are digested by this lysosome. The lysosome, it consists of certain enzymes which can digest these foreign particles. That is the main function of this lysosome. In certain circumstances, in specific such a situation, like when a cell is damaged, if this damaged cell exists in the tissue, it will spoil the environment. So when a cell is damaged, these lysosomes burst and they digest the contents. They clean up the area. So that is the major function of the lysosome, is to digest the unwanted materials and to keep the environment clean. Environment in the sense the tissue, the cell, the external and internal environment of this cell. It will be kept clean by the lysosomes by digesting the unwanted materials with the help of digestive enzymes they consist of, and these digestive enzymes are prepared by rough endoplasmic reticulum. So that is the reason the lysosomes are called as suicidal bags of a cell, because when a cell is damaged, the contents, the components are digested by this lysosome. It is called as a suicidal bags of cell. So when the cell has to be digested, these lysosomes, they burst and they digest the cell. That is the reason they are called as suicidal bags of the cell. So this is about the lysosomes. So the next cell organelle is mitochondria. Mitochondria are generally called as powerhouse of a cell. Powerhouse of a cell. Why they are called as powerhouse of a cell? Because they help in the generation of power. Power. In terms of general uses, what do you mean by power? In our house, we call the electricity as power, because we need electricity to run every machine, appliance in our house. TV, refrigerator, whatsoever, it runs on electricity. So we call the electricity as power. But whereas in case of our body, the different reactions takes place in our body, the cellular activities takes place in our body with the power in terms of ATP. The power in the form of ATP is required. So we need energy. We get the energy by taking food. Ultimately, the food has to be converted to which form? ATP. What is this ATP? A molecule named as adenosine triphosphate. This is ATP, adenosine triphosphate. So this is the energy packed molecule present in our cells, which is helpful to make new compounds, helpful for mechanical reactions, chemical reactions, whatsoever. If you have to lift something there, your muscles have to be contracted. For the contraction of your muscle, ATP is required. If you wanted to talk, your muscles in your jaws must be moving. The muscles of your throat must be controlling the airflow. For these muscles functioning, ATP is required. So that is for any kind of physical work or chemical work in your body, energy is required in the form of ATP. So this ATP is considered as currency of your cell. Currency of your cell is nothing but this ATP. So this kind of ATP, as you have come to know the importance of this ATP, such an important ATP is generated in mitochondria. Of course, ATP are even generated in cytoplasm, but major part of ATP are produced in this mitochondria. So we know whatever the food we take, it gets converted to energy. The main part of this conversion takes place in the mitochondria. So that is the reason it is called as powerhouse. So that is the function of the mitochondria. Let us see the structural features. Mitochondria is a membrane bound organelle. It is having two membranes, outer membrane, which is very porous, inner membrane. You can see the picture how the inner membrane is. It has got several folds. So why several folds? That is to have more surface area for large number of reactions to take place. Large number of reactions takes place at this point on the surface of the inner membrane. So it has got two membranes. This is the mitochondria. Mitochondria are rod shaped, sometimes oval, sometimes circular, sometimes comma shaped, spherical shaped. In different shapes they exist. But they exist in each and every cell, that is to produce energy. In all the eukaryotic cells we find the mitochondria. Now, this mitochondria, it is the place for many metabolic, biochemical reactions, and the other important distinct feature of this mitochondria is having its own DNA. It is having its own ribosomes, so by that it can prepare its own proteins. So mitochondria is able to produce its own proteins itself. So that is the structural features of a mitochondria. And if you look at its function, the main function is to generate power. In which form? In the form of a chemical molecule called ATP, that is adenosine triphosphate, which is called as currency of our cell, because ATP is very essential for every reaction, whether it is a chemical reaction or any kind of a mechanical movement or whatever. So for that, energy is needed in the form of ATP. So that kind of ATP are generated in this mitochondria. So the other cell organelle plastids. Plastids are found only in plants. You do not find any plastids in animals. So they are only found in plants. Plastids are basically two types. They are chromoplast and leucoplast. Chromoplastids and leucoplastids. Leuco in the sense white. So which are white plastids? That means colorless. Chromo means colored. Colored. So as the name suggests, they are colored plastids. These chromoplasts, example is chloroplast. So these chloroplasts are of different colors, green, depend upon the pigment, red, yellow. So these chloroplasts, some of them are green, red, yellow, depending upon the pigment contains. The chloroplast, if it contains chlorophyll, which is a green pigment, chlorophyll, they are green. They contain if some other pigment, red color pigment, they are red in color, yellow in color. We see the fruits and vegetables in different colors. You can see the capsicum in red color, you can see the capsicum in green color. It's because of the pigment which it contains, the chloroplastids. So chloroplasts are a kind of plastids which contain color pigments and appear in different colors. Leucoplast, the leucoplastids or the leucoplast, they contain stored starch or oils or fat and other materials stored here, no pigment. They are the storage bags. These plastids, which are white leucoplasts, are the storage bags of starch or oils or fats. Whereas the chloroplast, they contain different colored pigments and they appear in different color. If you see the structure of a chloroplast, the chloroplast is also a membrane bound organelle, just like mitochondria. There are certain similarities with mitochondria. If you see the chloroplast, you can see the membranes arranged in stacks which are called as grana, and these grana are fixed in a material, embedded in a material called as stroma, and these grana are interconnected like this. And the chloroplast, which contain chlorophyll, they are green in color and carry out a process called as photosynthesis, preparation of food, photosynthesis. So that is the structure of a chromoplast, that is a chloroplast, the plastid which contain the membranes in the form of stacks. As like in mitochondria, the plastids also contain their own DNA and own ribosomes, which is absorbed in even in plastids. Vacuoles are the small sacs present inside the cell which consist of materials like simple sugars and other storage materials. Generally, we see vacuoles in large size in plant cells, especially in plant cells. They occupy the space from 50% to 90%. If you see a plant cell, just this is a plant cell. If this is the cell, it may contain this much size of vacuole which is occupying more than 50% of the space. In most of the cases, the vacuoles are filled by cell sap, which gives turgidity and rigidity to the cell. And most of the important materials that are required in a lifetime of a plant are stored in the vacuoles, like amino acids, some important organic compounds. They're all stored in the vacuoles. In some cases, certain waste materials are expelled out of the cell by the vacuoles. If you see the case of amoeba, in its body, whatever the food it is taken inside its body, the food is stored in the form of in a vacuole called as food vacuole. So the food vacuole is the only place where the food is stored and it is digested even. And when the waste is about to excrete, you call it as contractile vacuole. So in this way, the vacuoles help amoeba in taking the food, digesting and excreting. And if you see in plants and animals, the vacuoles, they store different kinds of materials, especially in plants. Food materials are stored in reserve in the vacuoles. And plants, they also store important organic compounds, materials, amino acids in the vacuoles in their cell. If you see the plant cell structure, definitely you will find the vacuole taking place large space and which is filled with cell sap, which provides turgidity and rigidity to the plant cell. So that is about the vacuole. So these are the various kinds of cell organelles which we have learned in this unit, the fundamental unit of life. Here we studied about the structure of the cell, the main components of the cell membrane, cell wall, nucleus, cytoplasm, and we studied about cell organelles like mitochondria, Golgi complex, endoplasmic reticulum, ribosomes, lysosome, and how these cell organelles are helping in the functioning of a cell.